So hi everyone, I'm Kwasi, and today I'm going to introduce you to the various microscopes at the Harvard Center for Biological Imaging and what are the applications that you can use for your research. I'm going to briefly give you an overview about the various techniques, how they are used, and what microscopes we have in the facility that apply those microscopes, and then give you a guide that will help you to pick the right microscope at the center. So for every imaging experiment that it always begins with sample preparation in the laboratory, where you determine what samples you have. Is it going to be a full tissue or a whole organism, or the cells will be growing on just a cover slip? Next are what are the fluorophores you'll be picking up, and then the type of mounting that you'll be using. And I will describe that in details later on. Next is the application of the various microscopy techniques. That depends on first, what are the chamber? Is there a chamber on the microscope or not? And if there's a chamber, is this good for live cell imaging, or it is used for other purposes? Second are the objectives. What magnification are you interested in imaging your sample? And more importantly, which very few people think about, is the resolution that that objective is going to give you. The third thing is what lasers are on the microscope or LEDs, and how are these going to be detected once you eliminate your sample. The final piece is get my picture off. Yeah, and the final part is image processing. For today, I'm not going to talk anything about the image processing, but that is very important. Getting your initial image, as you can see here, is not sufficient. You might want to do subsequent processing in order to get a better resolution or a better image quality. All right. So the major question that we usually get at the HCBI from users is are classified into these three broad areas. First is the sensitivity. Will I be able to detect my signal? Is there enough signal to noise ratio? And do I have enough, is the detectors good enough to detect that signal? The second thing is resolution. Everyone comes up and says, well, you know what, I want to get a very high resolution image. And what are the things you need to consider when you think of a high resolution image? And then the final part is the speed. And again, there are two things people think about in terms of speed: the acquisition speed, how fast can I acquire the image? And two, can I be able to scan multiple places and be able to capture that image within a short time? Okay. So the big question is, which microscope will be able to do all these, get me all these things, and even do more things for my experiment? And it's always a trade-off. As you can see here, it shows a triangle. That means that as you try to get high sensitivity, you may be sacrificing a bit on speed. And if you get very good speed, probably you might be sacrificing on the resolution. So there may be a trade-off between these three things as you set up your imaging or acquisition experiment. So let's take the microscopes one by one. The first microscope, or the general microscope you're all familiar with, is the Widefield microscope. So with the Widefield microscope, you have a light source, and that light source has an excitation filter, and it pick one wavelength that goes through a dichroic mirror. It's reflected onto your sample, and then if you consider this line as your focal plane, then you have an illumination of a wide area. The out of focus scattering that comes here are all collected through your objective, and that is detected on your camera. Okay, so in a wide field process, your major issue becomes the out of focus scattering that we are going to capture on your detector. However, if your sample is very close to the cover slip and it is very thin slice, you are more likely to capture a very good focal plane right at the cover slip. And that is something you would have to consider. In the confocal mode, though you have such a broad focal depth, you are just illuminating or capturing signal from, so all the signal that is coming from the out of focus area, they will all be blocked by the pinhole. So here you start with a laser, you illuminate your sample, and then the emission that is collected, the out of focus are all blocked, and then the focal plane is the one that is detected on your detector. And here's a PMT, okay? So I'll discuss a little bit about the differences between your camera and then your detectors. And then in the multi-photon situation, again, you are using a laser line, and then as you illuminate your sample, you illuminate only the focal plane, and that focal plane is what is eventually collected on your detector. And then the final part, the final system that I will address is your light sheet. In a light sheet configuration, the light, your laser comes out straight onto your sample. So you illuminate your sample as a sheet of light, and then that is collected straight onto your camera, okay? So if you compare your Widefield and your light sheet, they are both using the Widefield technology. And then the laser scanning and the multi-photon, well, here there's a pinhole that blocks the illumination. And then in this particular case, there's no pinhole. So you collect the sample straight from the focal point. So taking the various species one by one, first is looking at the differences between your Widefield LEDs and then your lasers. So in terms of Widefield, you have your LEDs in this box, and these LEDs can all combine and give you a white light. There may be an excitation filter that will select a particular wavelength, and then you illuminate your sample, and then you collect your emission into a detector right up here. You might also have a situation where you can have your white light, but because you are getting a very good signal, you would just collect the wavelength right from that particular excited LED. The configuration that is becoming more and more common nowadays is you being able to illuminate different LEDs, and then with or without. So as you can see from here, you may be able to remove your excitation, the filter, and be able to collect your sample through, and that is individual illumination of your LEDs. Okay. The other situation that we have is when we think of the confocal mode. So in the confocal mode, we have all these types of lasers, starting with the 385, which we don't see here, all the way to the 640, and now you can even go to the far red. And the type of lasers that we have, and then the final part is the detection that you can observe. So for instance, we take a 405, you can be able to image a DAPI, a BFP, or a fused. And in the standard traditional microscopy, most people have 4 dyes, where you think of your 405 for DAPI, then you have your GFP, which is in the 488 line, then your 561, which is in the red, and then your far red, which is the 630. These are the 4 standard, but there are techniques which I will discuss later of how to be able to image when you have things that are in between these various laser lines of, or you have fluorophores that overlap in terms of their emission wavelength. Okay. So in terms of the detection of your signal, as I said, we have the Widefield and a Confocal. In a Widefield mode, you are mostly either using an EMCCD or a CMOS camera. The EMCCD had been what had been used for a very long time, and now the CMOS camera is catching up. Initially, the CMOS was barely used, and the major reason was that the quantum efficiency was low, even though they have a very good high frame rate. In terms of the EMCCD, they have a very high quantum efficiency. That means they are sensitive enough to detect a signal, but then the scanning were slower. New systems in the market now have improved quantum efficiency. So you'll be thinking of something along the range of about 80% or higher. So, but then the frame rate is always higher, and therefore it is becoming the choice for most microscopy applications. When it comes to your confocal mode, you are using detectors, and here the detectors are based on a photocathode array, whereby there's different voltage that goes across your camera and that detector, and that helps to determine the sensitivity. Various detectors have various quantum efficiencies. So as you can see from this diagram, the ultra bialkali detector can detect things better in the UV range. The GASP detector is better in the red and then green wavelength. And then there's another multi-alkaline detector, which is having a better sensitivity in the far red. So again, during acquisition, you want to have your emitted light being collected on the right detector in such a way that you'll be able to distinguish, be able to get the highest sensitivity based on the detector that you have. All right, so how do we implement these ideas at HCBI? On the booking website, here is a representation of the things that will help you to determine what microscope to choose and how to pick the data, how to pick the right one. So here's an example of a system which is a cell discoverer. It has been indicated to be for live imaging and then high content screening. The reason why it is for live imaging is that you can see there is a chamber that is around the microscope, which means you can isolate and have a better incubation system in there. Okay, next is a little description that tells you about the various mounting system that can go in, and then, so here is a dish. There's also up to 300 web plate that can be used in the system. There's a CO2, there's humidity, and then heating. So that is indicated there. And here are for me, this is the section I find very useful. And the reason is that it tells you what are the LEDs that are available, and based on the fluorophore that you have in your sample, you'll be able to tell which one would be ideal to use. Next is the filter set that you have in to be able to remove any excitation light that may come through, or signal that will be resulting from other emission that is being collected from different parts of your sample. For instance, if there is a breakthrough that is coming from one pass to the other, this set of filters is going to help to separate or remove them. We have right here, we have given some ideas of what are the best options to pick in order to separate your dyes uniquely. So here is for DAPI, then you have the GFP, the red, and then the far red. So it tells which combinations you can separate very well. Okay, so that is using a wide field system, as I said, this is a wide field because you are using all the LEDs in the process. Next is a confocal. So here's another example of a confocal with a flame feature. It tells you a bit about what wavelengths are available and what detector that we have for different applications. Next is the laser lines that are on it. So you can see the various laser lines and how to apply them. And then there's the description of the various dyes that you can image using the HAT film. So once you have an idea based on which microscope, what are the various laser lines, the next challenge that comes up is, well, how do I resolve and get a high resolution image? And here is a short description of what to think about when you are looking at resolution. So in resolution is mostly different from the quality of the image. So as you can see from this diagram, the image is not very good. So you can easily see it doesn't look sharp. It doesn't look, it looks a bit faint or blurry. I intentionally took this image to make this point that the image may not be sharp enough, but will we be able to discern between two different spots? So on the left, as you can see here, you are able to distinguish between these 2 points because they have the separation of that area. There's a significant separation. As they get closer and closer, it becomes more and more difficult to resolve them. And that is what, in many cases, when people are asking about resolution, that is something you have to think about, that the idea is to be able to separate your two different signals and be able to distinguish between them very well. Okay. Next, how do resolution and then the objectives interact? And that is an issue that it is not easy to explain, but I hope this diagram is going to help us with it. So in many cases, for each objective that you have, from a 10x all the way to a 63x, beside the objective magnification is the numerical aperture that is indicated right here. And historically or traditionally, as the magnification gets higher, your numerical aperture also gets higher. Okay. So there is this proportionality relationship that tells you that as you get to higher magnification, you are going to get a better resolution, and therefore you'll be able to separate your various features in your image. However, with time, this is no longer very true. An example is what we have here with a 63x water objective. So this 63x water objective has a resolution, has a numerical aperture of 1.2, but then the 40x objective here has a numerical aperture of a 1.3. Therefore, if you want to discern two different spots, you are more likely to get a better resolution and be able to distinguish between them with a 40x oil versus a 63x 1.2. And this is something that a lot of people find difficult to accept because of the traditional concept that the higher the magnification, the better the resolution. And therefore, in thinking about your resolution, it is always important for you to look at the numerical aperture of that objective and let that guide you in determining the resolution. If the idea is to get a zoom-in image, yes, then the magnification is able to deal with that. And the plot of that concept, here's an illustration of that as the numerical aperture increases, your resolution increases from, here is maybe, I'll put it, it's an increase in resolution because you are dropping in terms of how can you distinguish between two units which are in a very close proximity to each other. So right up here, about 250 or 200, you'll be able to resolve two objects as your numerical aperture increases. And at the same time, as you change your frequency or the different wavelengths, they all don't respond the same to the numerical aperture. So a typical example would be taking any of these wavelengths. So when you are in the ultraviolet or in the DAPI range, your resolution is far better than when you move to a green or to the far red. So this is something to also know, that you are not getting an equal resolution, even though you are in the same focal plane, because of the differences in the wavelength that you are using to capture that image. So what are the microscopes that we have at HCBI, and how can we be able to apply them? So the first is the Widefield microscope. As I said earlier on, you are going to capture everything, including the scattering light that comes that are captured in the epifluorescence mode. Usually, your sample thickness is less than 50 micron. It must be close to the cover slip, and the Widefield technique, it works better for monolayer of cells, tissues, and then fixed and live samples. So here are some sample holders that can go on that microscope. It can take a slide, a multi-wear plate, and then dishes. On the LSM 900, the new microscope that we have, there is a camera right here, and that camera is what you'll be using for the Widefield imaging. And in the Widefield mode, you'll be able to run a transmitted part. I've not discussed much about transmitted mode because very few people use are interested in transmitted imaging, but there is a bright field, a DIC, and a phase contrast mode. And then we have the fluorescence mode, which I just described. When you are very close to the cover slip, you'll be able to capture the image because of the thin slice. Again, to be able to improve the resolution, or to improve the quality of the image, not the resolution, the quality of the image, a deconvolution or further processing is required to make sure that the image is sharper and remove all the out of focus light that is associated with the wide field imaging. So to get rid of the wide field, the out of focus light that comes with the wide field microscopy, the first is using the software, going through deconvolution, look at your point spread function, and then use those values to be able to process the image from a broad or a more blurry image and make it look sharper. However, on some of the wide field microscopes that we have, there is an Apotome mode, and the Apotome is a grid that is put in front of your image, and that grid can be used to back calculate and then remove all the scattering lights that are associated with your image. So in cases where your sample is very thin, they are close to the surface, you want to get a high quality image, but you don't want to run through the confocal, here is a good example of using the Apotome mode, which can help to remove that scattering and make it better. Two of the systems, the HCBI, the Axial Zoom, and then the Lyra have the Apotome that you can use in your imaging. Next is the confocal microscopy. So here we have the LSM 700, 880, 980, and then 900. They can be used to image anything right from the cover slip to about 200 micron into your tissue. Again, they can work with monolayer of cells and then tissue slices, and these are the chambers that can be used on these systems. So here are some examples of images that have been acquired using these microscopes. So we have the confocal mode, which again, the resolution is about 250 nanometers. And then there is the Airyscan, which can then be able to distinguish objects that are even closer than the 250 nanometers. And I will discuss this in details later on. Then we have the flame, which is on the 880. And the goal of the flame is to be able to look at your signal duration and be able to characterize the properties of the signal that you are capturing. And then the final part is the confocal spectral and mixing process, where you can distinguish between dyes that are extremely close to each other. As I said earlier on about the different laser lines, it is we are traditionally limited by how much limited by how much we can distinguish between dyes when they are so close because of the breakthrough feature. But with spectral mixing, you can be able to distinguish between as much as 6 different dyes in one image. And here is an example from one of our users, Xiaomen. So here you can see that we have about 6 different fluorophores, and then she's able to distinguish between them in this image. Next is, what is the advantages of moving from a confocal to the Airyscan mode? So in a confocal imaging, you capture an image, but because of the diffractor limiter spot, you are still restricted to 250 nanometers. In the Airyscan, using the Airyscan technology, you can be able to resolve them even further, and that helps you to distinguish. Probably it's not clear from here, but you can see that there's a small gap between these two signal points, and it's difficult to distinguish that from here. So the idea of using the Airyscan is to be able to improve the resolution, and then, as I said, resolution and then the sharpness are related, but that gives you that structure. So here is a quick representation of why the Airyscan is faster. First, instead of scanning only one region at a time, you can multiplex them. And by multiplexing and scanning across, you can be able to increase first your resolution. You can increase your resolution from the 240 to 120. Increase the speed at which you are scanning by even a factor of 2, and in some cases 10x, 10 times that the speed that you have. In general, for live cell imaging, the Airyscan is better because you can scan faster and be able to capture your images in a more rapid manner. Next is the ELIRA super resolution system. So on the ELIRA, you have anything that is mostly less than 50 micron. That would be better for the ELIRA. And you are looking at samples that are either a monolayer or fake samples. Again, they are the chambers that can go into the ELIRA, and there are different modes on the ELIRA. We have the same. So with same, you can get almost up to 60 nanometer resolution using the same squared system. And here's a comparison between the same and then the Widefield image. In bacteria, where you have the thickness of a bacteria to be around 2 micron, it is always a challenge to be able to see subcellular structures within the bacteria. And again, with SIM, and then using the SIM squared process, you can be able to resolve even tinier things within the bacteria. The other technique using the ELIRA is the single molecule localization microscopy. And here's an example of a wide field image. And then with the single molecule localization, you can be able to detect even smaller objects that are way beyond the diffractal emitted spots. So any application between wide field all the way to the super resolution microscopy, you have different applications and different microscopes that can help you to distinguish your data, distinguish and improve the resolution of your features that you're looking for. Okay. Next is the high content screening microscopes. The first is the cell discoverer. The cell discoverer, again, is a wide field system. It's able to image and anything below a 50 micron in thickness. It's good for live and fixed, and then it's automated, and you can run both a time lapse and multi-position. So here are a few of the different systems that plate or mounting types that can go into the ELIRA, and then you can be able to run multi-position. So in terms of multi-position, imagine you have your cells all loaded, or you are doing an assay in which you have treated all these different kinds of wells. With the ELIRA, you will be able to set up a system to be able to image everything and automatically without you being there to cherry pick and decide where you want to scan. Okay. It can also run both a fluorescence mode and then a bright field mode. So in a bright field mode, you have samples which are either non-stained or stained with a dye which has no fluorescence. You will be able to detect that that sample using the ELIRA. And I'll give an example in the next slide. So in the next slide is the Airyscan. Again, this is a wide field, but it is an upright microscope. So you are imaging right from the top. The objectives are at the top. It is strictly for slice, and this is because you load a slice right in here up to 100 slice, and then it has an automated way to image your slice and to capture the label as well. And as I explained earlier on, the difference between the fluorescence and then the bright field mode is that when you have your fluorescence, you have to excite it with light and then capture the emission. In your bright field mode is just a dye that is staining your tissue, and you can shine white light and capture the variation or dye that had labeled your tissue. And the third wide field microscope that I'll discuss is the axial zoom. For me, it is one of the instruments that I find as very useful, but less used. And the reason is that it has a very unique feature in which your zoom, it is an optical zoom that is happening within the tube lens here. You load your sample onto the stage. It could be a dish, a slide, or if you don't even have a holder for it, you can put the object right on the stage. And then using the optical zooming, you can zoom in and capture, again, both fluorescence and then the bright field. Okay. So let's move to more specialized systems that we have. The first is the multiphoton microscope. It is an upright microscope. You can use it to image tissues, organs, and organisms. If your tissue or your sample is cleared, your ability to image deeper into the tissue gets better compared to the uncleared tissue, but you still have a range up to 6 millimeter that you can image through your tissue. And the sample holders we have, you can use a slide as before, and then we have these dipping objectives. And because of the diameter of the dipping objectives, we always recommend a 60 millimeter dish or anything higher than that. That will give you the ability to image directly through your through your buffer or your media. We have both dipping objectives and clearing objectives that will allow you to be able to image your sample. So using the multiphoton, you can do a 2-photon imaging. There's a second harmonic generation, which can look at collagen and other things. And then there's the confocal mode, which is that single photon application of excitation and then emission. So you have the ability to use any of these approaches using the 2-photon. The next specialized microscope I will discuss is the light sheet. So the light sheet can image even a higher area or a higher volume. So here you are looking at something within less than 10 millimeters. It is ideal to use it for cleared tissue. Most of the time we use it for cleared, but we can still use it for uncleared, but it has to be super transparent. It has to be transparent for us to be able to capture that image. Now, how do you mount your sample? So there is a sample holder where you can glue your tissue straight to the sample holder and image it and put it right in front of your objective for imaging. Or there is a capillary tube that you can put your sample in and then shine the light sheet and then image it through that. Okay. And with that approach, you will be able to image a whole organism, a whole tissue without any slicing. So I know we have been used to slicing tissues in order to get, be able to image deeper into our tissue. And that has its own challenges of now putting everything back together to reconstruct that tissue. With the light sheet, you can be able to capture maybe an eyeball or a kidney or a whole brain without having to do any sectioning. The final or the next specialized microscope I will introduce is the palm laser microscope. It's mostly for isolating single cells. So if you're interested in isolating individual cells from a tissue, it is able to cut that piece, capture it on a plate for you, and then you can do subsequent analysis with them. And here is an example of how it works. You can map where the area you are interested in, you define that, and then the system can cut that segment uniquely for you to use for subsequent processing or subsequent experimentation. And then the final microscope, which is in the Austin Center, is the LSM 900. The system is an upright microscope. As you can see from here, it has both the ability to run a wide field mode and then also the confocal mode with using the detectors. You can, I've not talked about FRAB, but again, confocal and spectral imaging can be done. And then these are various applications that material science people can use. So in terms of the application, you can use it to look at surfaces. Depending on what features you're interested in the surface, the material science microscope can give you an idea of that. And the next person in the Lunch and Learn series, Chris, is going to talk more about the material microscope and its applications. So hopefully you all come back and hear a lot about how to use this microscope in your research. So here overall, before I sign off, this is what I think is a summary of what the microscopes at HCBI can give you and how it can help you with your research. So if you have your sample is less than 50 micron, a wide field microscope will be able to do both a bright field and a fluorescence microscopy. You can, if your sample is on a slide, in a plate, or in a dish, these are the microscopes that can be able to run those experimentations. However, if it is thicker than the 50 micron or up to the 200 micron, and you want to do confocal, frame, the Airyscan, again for fluorescence imaging, we have the LSM microscopes that can do a similar, can capture that image for you. For super resolution microscopy, you're looking at things less than 50 micron. You have SIM, Storm, TEF, Widefield, which are on the ELIRA, and that can take slice in 35mm dish. And then when the samples are thicker, up to 6 millimeters in thickness, we have the confocal, multiphoton, second harmonic generation. We have the 2-photon to use. And then the final part is for tissues that are quite large. You have cleared them, and you want to get the entire tissue or organ. The light sheet microscope is ideal. So I hope this guide will help you to be able to determine what microscope or technology that is useful for what you are interested in acquiring, and how the acquisition will be should be done. So the first part in my summary talked about the different microscopes, and then the second part of the summary tells you about at what level of data or imaging mode that you can use the different microscopy technology. So if you're looking at single molecules, here a Storm, a SIM, and a STED will be able to capture that single molecule for you. For people looking at organelles within the cell, again, a SIM, the STED, and then a confocal will be able to capture that. Organelles can, at the same time, can also, the Widefield can help, but again, it depends on how deep, how close they are to the cover slip. At the cellular level, we have the Widefield, confocal, and the SIM, and this can help with the, for you to identify that object. At the tissue level, Widefield, confocal, and then a spinning disk can do a similar thing. And then at the organ and organism level, you have the multiphoton and then the light sheet. So this is a very good way of looking at the various techniques, how they are used, and then what your research question entails. So with that, I'll leave it for questions and answers, and thank you for listening.